Hi guys, and welcome to another video. Today we're going to be reviewing the microcirculation of the papilla and the lamina in horses. It's a niche topic, but since there are diagrams available to us in Butler's textbook, we're going to review it in case it's ever on the certification exams. It might already be on them, I can't be sure, but I want to make sure this series is slightly future-proof as the exams evolve over time. If you're new here, hi, my name is Julia Walker, and I make videos going over the main topics that come up on certification exams for farriers. I'm attempting to make these certification topics more memorable and easier to learn, hopefully without making any factual errors along the way. And if you're not a farrier, that's totally okay too. I just like talking about anatomy and breaking things down, so hopefully this is helpful for you. But with all that being said, here we go. So first, what is microcirculation? Well, it's the small blood vessels that fill in a specific area. Think of it like a mini circulatory system. We'll be looking at the connections and relationships between the arterioles, or tiny arteries, capillaries, and venules, or tiny veins, in the papilla and lamina. The two main diagrams we'll be referencing are in Butler's P3 on pages 621 and 622, sort of the main reference for all of this. They're great diagrams, but the laminar diagram is kind of complicated, so we're going to try to break that down today, as well as the papillar diagram since it's related. We have to quickly recall that the papilla are the tiny cone-shaped structures that cover the coronary corium and solar corium. So that's kind of these, they kind of look like hairs up top and down below. They are the foundation of the hoof wall and horny sole, creating tubular and intertubular horn as they keratinize. Check out my keratinization video if you want to learn more about that. The lamina, specifically the sensitive lamina, are the soft ridges that cover the dorsal surface of the coffin bone and interlock with the insensitive or horny lamina to create the bond that connects the coffin bone to the hoof wall, like a biological suspension bridge. Lovely. Starting with the papilla, we'll go through and label this diagram. It's pretty simple, actually. So here we have the papilla. They form the tubular horn as they do their keratinization process. So kind of like this tube that comes off here, that's tubular horn. Between the papilla is still corium, which forms the intertubular horn. So there's kind of corium through here, and then this area in here is that intertubular horn. Basic stuff so far. Then we have the main artery and vein up here. So that's an artery in red, and that's a vein here in blue. And these branch off little versions of themselves into each papilla, consisting of a papillary artery and a papillary vein. So the red one here, that's your papillary artery, and the blue one here is the papillary vein. Great. You see they kind of swirl around each other in the papilla. And then off of these come the capillaries, which I have in kind of this purpley fuchsia color here. Which permeate most of the surrounding tissue. And then between the papillary artery and the vein, there are AVAs connecting the two in case of emergency. That's kind of that teal color. So there's an AVA. And there's a few of those in each papilla. And here we have a label diagram if you want to save it for your studying. It's not a lot different than the diagram in our textbook, but it is color-coded and with fewer crossing lines, so if that's helpful for you, here you go. Now we're going to break down that laminar microcirculation diagram. This diagram was the one that originally freaked me out a bit when I saw it first and made me say that I didn't understand the circulatory system at all. So if that's you as well, you're not alone. However, we're going to break down what's going on in several images so that you can actually understand each piece that makes up that total image. And to do that, we need to have a good idea of how we're looking at this situation. So here's the direction that we're looking at the lamina. It's kind of like we climbed in between the sensitive lamina and are looking at them from the side. I guess that would mean we're kind of looking at the sensitive lamina from the viewpoint of the insensitive lamina, but hopefully here you have a good idea of the direction that we're looking at. So this is the image from the title screen, which even when it's color-coded is a lot. I used the same colors I've been using for all the structures so far, so feel free to take a moment to let it soak in. The main structures we'll be introducing to you today are the parietal artery and vein, the axial arteries and veins, interconnecting arteries and veins, and by association the AVAs, the marginal vein, the peripheral arterial loops, and the abaxial capillaries. Sound good? Weirdly enough, by the end of this, you'll probably be able to distinguish them all. So you've got this. Okay. Now, you heard me mention the words axial and abaxial back there, and if you already know what those mean, fantastic. For the rest of us mortals, you've probably only heard those used by vets or seen them in the textbook here and there and just assumed that those were words that you weren't really going to have to know. But I promise it's super simple. If you were a horse and you were looking down at your front hooves like this, these are the two directions. As a direction, axial means towards the central axis, like how the earth spins on its axis. It's the central point of the limb. The medullary cavities and the bones, for example, are axial to almost every other structure in the leg, so that's easy enough. The term abaxial basically means away from the central axis. So the periopal is, for example, abaxial to the rest of the structures at the coronary band. The AB part basically means away from. Sort of how the word abnormal means outside the realm of normal. So you can throw outside in there too. Wonderful. So let's start with the arteries. Just to make sure we're on the same page here, on the left you have the insensitive structures colored in hoof color so you don't get lost. See how it says out that way. On the right are the sensitive structures going in towards the coffin bone, so P3 lives somewhere over here. And in front of us, this is a side view of the lamina. In the arteries, we know that the blood is flowing away from the heart, so we can say it's going from right to left in this diagram. So blood is going this way. Great. The first structure we need to label is the main artery for this particular lamina, called the parietal artery. So here we go. Parietal artery. The word parietal means that it's a part of the wall of either the body or a structure, so think of it kind of being at the boundary of the lamina. So I'll write that here. Cool. Then we have the axial arteries. So these are the axial arteries. Here and here, all the ones going in that direction. Cool. And there's just several of them going away from the base of the lamina. Then we have the interconnecting arteries. Which do exactly what you'd expect. They're these ones. They are connecting everything in the middle, and they get the blood flow down closer to the cellular level. Out closer to the tips of the lamina, loops connect the axial arteries to each other, kind of like the terminal arch inside that coffin bone. That's what arteries like to do on their ends. And these here are called the peripheral arterial loops. They're kind of out on the outside, sort of like how your peripheral vision is on the outside of your frame of vision. Awesome. So that's what the arteries do in the lamina. There's that parietal artery, that main one, the axial arteries shooting outward, the interconnecting arteries connecting everything, getting the blood closer to the cells, and then at the ends of the arteries we have these peripheral arterial loops. Wonderful. And here's a proper labeled diagram of the arterial structures in case you want to save it. A little more legible than what I just did. Now we have the veins. It's a super similar structure. There are just more interconnecting veins and a couple other differences. Here the blood is flowing from left to right. So going back this way, back up towards the heart eventually. So in the opposite direction. On the left side, the most abaxial structure in our diagram total is a vein that runs along the tip of the sensitive lamina, and this is called the marginal vein. Sort of like how I'm writing marginal vein in the margins of this slide, off to the side. This dumps into the interconnecting veins, which same name as the arteries, right? which are pulling blood up from the cellular level as well, and then channeling back into these main veins here, which have the same name as the arteries did, which are, if you remember, the axial, except this time they're veins. And there's just a series of those. And then the axial veins then flow back out of the lamina. When they reach the base of the lamina, they meet up with the main vein that will take blood back up, which is called, same as the artery was, the parietal vein. And boom, that's all the vein action going on in the lamina. So here we go, labeled the 4 main venous structures in the lamina. You got the parietal collecting vein, the marginal vein right on the tip of the lamina, got the axial veins going in between, and then the interconnecting veins going between the axial veins. Excellent. Next up, there's a kind of fun structure in the diagram that sneaks in almost unnoticed. Those are the long capillaries that run up and down the lamina in pairs like a weird ladder on either side of the arteries and veins. Those are called the abaxial capillaries. And with that, we only have one structure left to learn about, and that's an old familiar friend of ours on this circulatory system series. But I'm going to warn you to show them. We're going to go back to that combined diagram because they involve other things. So this feels simple, no longer simple, right? But here we are, lots of squiggles. In fact, extra squiggles in bright blue, and those extra bright blue squiggles are the AVAs, or arteriovenous anastomoses. All these little dudes, and you can see that what they are doing is they are connecting an interconnecting artery here to an interconnecting vein there, interconnecting artery to interconnecting vein, same, and on that diagram in Butler's, you'll see that's where they are touching. They are also more sac-shaped than corkscrew-shaped in reality, but I figure it's important that you can see that there's something special instead of just drawing a blob. But think of them kind of like balloons or something like that. And that's it. We have the AVAs, you've got the interconnecting arteries and veins, you've got the axial arteries and veins, we've got the marginal vein, and in red we have the peripheral arterial loops. Over here, kind of with the same name, the vein and the artery are both those parietal veins and arteries. And over here we have these abaxial capillaries because they're running up and down away from the center of the limb. Great. And those are really the main structures that we need to know about. So the last thing on our to-do list is to review. Like I said, the reference diagrams we're breaking down today are in Butler's P3 on pages 621 and 622. So please go check those out and see if you can make sense of what's going on there after going through this. But let's do some review questions. First, are the lamina axial or abaxial to the coffin bone? Thinking about those directions, in this case, the lamina are away from center relative to the coffin bone, so that makes it abaxial. Is the endosteum axial or abaxial to the periosteum? So if you remember the anatomy of a bone, the endosteum is the lining around the surface of the medullary cavity inside the bone, and the periosteum is the lining of the outside of the bone. So if the periosteum is outside the bone and the endosteum is inside the bone, the endosteum is axial to the periosteum. Next, the blank vein runs along the end of the lamina, forming essentially the most abaxial circulatory structure in the lamina. Do we remember the name of that outermost vein? Well, it is called the marginal vein. Then we have the blanks lie just axial to the above vein. So we're talking about that marginal vein now. So just axial to that, when we're looking at that diagram, what was the structure that sat there, just inside of the marginal vein? It's the structure at the end of the arteries. Remember those loops? Those are called the peripheral arterial loops. Remember, peripheral means along the edge of, like how our peripheral vision is along the edge of what we can see. Excellent. Lastly, AVAs connect the blank arteries and veins. Do you remember what types of arteries and veins are connected with those bright blue squiggles? They were the small ones that filled up most of the space, which are called the interconnecting arteries and veins. Awesome job. You did it. You made it through. Fantastic. Hopefully you learned something. I know it's a lot, but it'll give you something to think about the next time you look at a hoof. Imagine all those tiny blood vessels and try to remember their names. For now, we are all done with the circulatory system. Great work. Next up, after consulting with my mentor, I've decided the next video series ought to be the tendons and ligaments series, since apparently that can be a big sticking point on the exams, so get ready. First up, I'm going to map out the origins, attachments, and insertions of the main tendons of the limb and the suspensory ligament, and hopefully come up with some good visuals for you guys to help remember things. If you already know it, great. I'll have other videos in this series talking about more details about function and types, the ligaments of the fetlock and pastern, the tendon and ligament problems that come up, and if you have any other topics you'd like to see covered in this upcoming series, please let me know in the comments. If you have any corrections for the video today, I would also love it if you let me know down below, or any questions related to this topic. Hopefully these facts are at least fairly straightforward. I know it's a little more complicated than most things. But if you know about any new research that's been done on the microcirculation in the lamina or papilla, please share a link with us in the comments. I'm sure everyone would love to know about it. This is accurate as of 1990, but it's a pretty interesting hot topic right now. In the meantime, I'll keep cracking away at the next topics. I'll try to get the next video out as soon as I can. Best of luck studying to you, good luck on your exams, and as always, stay tuned for more.